Hey everyone, this episode is long overdue. I think I recorded it with Dr. Schwartz 2 months ago, and I'm getting to it now, which is kind of my fault. But anyway, this episode features Dr. Schwartz, who is video calling from Michigan. Dr. Schwartz is a professor of micro and molecular biology at Michigan State University. He received his BA at UC Irvine and his Ph.D. at MIT. He also received his postdoc training at UCSD, CalTech, and UCLA. The study we're discussing focuses on endocrine-disrupting hormones and environmental exposures, their effects on mammary tumorigenesis, and how their behavior shifts depending on the diet they're paired with. This episode is going to make a lot of references to a study published in 2020 that looks at the diet-dependent effects of oxybenzone on tumorigenesis, so I left a link to the study in the description. Thank you for clicking on this podcast, and I hope you enjoy it.
You having a good day,
Um,
Tanish?
yeah, the weather's kind of crappy, but that's pretty much it. It's okay
We
today.
have
How about
smoke in
you?
Canada.
Yeah, we had that. New York was pretty bad.
Yeah,
I live
today
close to
was
New York,
horrible.
so that was pretty bad.
Yeah. I mean, it actually, it's amazing. You don't usually think of it as being above us when these huge clouds of smoke, drift. But in fact, you can smell it in the air, you know, and that's a little disconcerting
It
because then you
was,
know you're
yeah,
breathing this stuff.
yeah. We were in school when it happened, and like
Mhm.
it was like orange outside, so everybody wasn't focusing because it was like this orange hue in the clouds, and it was reflecting onto the classroom, and it just looked almost
Yeah.
like apocalyptic. It was cool, but also kind of scary, you know?
Well, luckily it was not orange today here. You had it
Really?
worse, but it was enough that it just smelled like a smoky room does. Um, you know, the day after people have been someplace and they've been smoking and
Yeah,
they're out of there, but it still smells kind of a stale, moderately offensive odor. Could be
yeah.
worse,
That's
but better. So, okay, yeah.
just like get started. Could you like give an introduction on yourself and what you do right now?
Yeah, so my name is Richard Schwartz. I'm a PhD. I got my training officially in biochemistry from Massachusetts Institute of Technology. I'm a professor at Michigan State University. I've been there now for about 37 years. Um, and besides administrative and service and teaching responsibilities, I've run a laboratory that whole time. And over the years have been a researcher in the area of both cancer, mainly cancer immunology, and also studies of inflammation. Those would be my two major areas. Um, what else can I tell you by background?
No, that was good. That was good. I want to talk about your study specifically in OncoTarget about
Hmm.
BP3 and
Yeah.
the diet-dependent effects of
Hmm.
BP3, and I want to make sure I just got something right first.
Oh, sure.
So this is very specific, but a low-fat
Any
diet
question's a good one.
A low-fat diet combined with BP3 had a lower rate of tumor development, but the tumors were more aggressive, so lower apoptosis and higher proliferation.
Hmm.
Is that right?
Hmm.
And then the higher fat diets had a higher rate of tumor development, but less aggressive tumors. Is that right?
I think, let's see. Um, yeah, I think that's a fair generalization. There's still some very aggressive tumors that form on the high fat diet, the adult high fat diet. Um, that would be um, similar to triple negative breast cancers if we wanted to translate from the mouse to the human. but the proportion of those tumors was a bit less than what we saw um, on the low-fat diet. And on the low-fat diet, there were fewer um, what we call differentiated epithelial tumors. Um, these are all pretty bad tumors that we get in this model system, though. Even the ones that are a little more differentiated in this particular model, they actually rapidly lose their receptors for estrogen and progesterone. So there, if I wanted to draw an analog to a human breast tumor, it would be um, like triple negative breast cancer, but a little less aggressive form.
Okay.
It's a little dangerous to draw direct
Yeah.
um, translations from the mouse system to a human system. Um,
Right. So rats are they more sensitive
or they're
to
mice. It's
the
actually
yeah, mice,
yeah
mice.
we yeah. Some people do use rats, in fact, in their experiments, but um, mice are smaller, and depending on where you head with a project, there are more opportunities for genetic manipulation of the animals.
Oh, that's interesting. I didn't think of that. So is that why you so what's the reason you use mice in this case?
Well, in this case, we chose mice just because, um, well, there are two answers. One is because we've been using the mice for quite some time, and we didn't want to change animals. We had done some purely dietary studies without benzophenone 3 um, in earlier years, and we wanted to use the same strain of mouse that we had used in our earlier studies. And that strain is BALB/c. It's the most common laboratory strain of mice. And the reason why we chose that particular strain of mouse initially is because when you feed it a high fat diet, it doesn't um, tend to become obese. It's somewhat obesity resistant. And what we were trying to understand was what are the effects of a high fat diet in the absence of um, obesity and in the absence of diabetes or metabolic syndrome. So, because it's confusing to identify what high fat might be doing to the occurrence of cancer if an
Yeah,
animal is suffering from some other metabolic dysfunction like
yeah.
obesity or diabetes or something like that. So it was a way for us to dissociate those kinds of comorbidities and get something that looked, it's not perfect, but something that looks more like a direct effect on tumorigenesis. So that's how we ended up with BALB/c mice.
So to keep like all the other variables constant to
Yeah,
make
we're
sure it
trying to
wasn't.
minimize
Yeah,
variables and minimize things that confound the interpretation. Like if just to draw a scenario, we found that high fat diet caused a higher incidence of breast cancer in the mice. So we wanted to be able to say, oh, it's the high fat diet of itself. It's not that high fat diet made the mice diabetic. And in the course of they becoming diabetic, that increased their susceptibility to cancer. We're trying to tease
yeah.
apart all these things.
Okay, that makes
And you
that's a good
always
choice
think
then.
about that when you use an animal system. Um, this is a little bit of a digression, but we like to use particular strains of mice or rats or any animal that we're going to use in these kinds of experiments because even though carrying some specific um, genetic traits that define a strain of animal, even though that means that you have to um, attach the caveat that Well, these findings may not be the same in another strain of animal. Okay. On the other hand, you have uh, with a homogeneous population, you're more able to identify effects. All the mice are going to behave to the environmental insult in a very similar fashion. So when epidemiologists are working with human populations, it's a real challenge. You need a really large population in your cohort to do the epidemiologic study because you have a diverse population of people. They will have different racial and ethnic backgrounds. They may have different economic backgrounds, which may change what they like to eat. Um, there's all these extra variables. So when epidemiologists want to do a really robust study, they need to enlist a lot of people to be able to identify relatively small effects, which many of these are relatively small
Yeah,
on a percentage basis.
yeah. So is it also true that the mice are mice more sensitive to tumor
Um,
development?
well, one thing we do with mice too is we can create a model system where the probability that the animal will come down with breast cancer is very high. So we don't need to study as many mice. That's another reason we use mice. When I say, well, there's genetic manipulations we could do. So the mice that we use over a course of um, 2 years, probably 90% of them are going to come down with breast cancer.
Okay.
Um, whereas in a wild type population that doesn't carry a predisposing mutation, the rate's really low. And that's another thing that makes epidemiologic studies really hard. If you want to study what happens among humans, how many humans do you have to enroll to see if there's a change in the rate of cancer? Thousands. And there's a real challenge. I mean, you have to struggle to set up epidemiologic studies because you want to confirm that what you find in an animal system is showing similar effects among humans. Um, on the other hand, it's another set of challenges that epidemiologists specialize in how to enroll the right set of people for a study and to track them prospectively over time and see what happens to them. Um, and it's done and there's some really great information that's resulted from those, but they can't do the kinds of experiments we do. You can't force, you know,
Yeah.
50,000 people to all eat some horrible diet.
Yeah.
um, and so all these kinds of studies actually, in the best of all possible worlds, we take what we see in the animal and we test it in a human epidemiological cohort, and sometimes we find things that they come up with, and we try to address them in an animal system and see whether, oh, this perturbation in the diet, what happens in this strain of mouse, what happens in that strain of mouse, can we change the outcome by altering things with the way it plays out by adding some drug to the system. So we go back and forth between human and animal models, and hopefully we arrive at a good picture of reality.
The human models are, I can imagine, a lot more difficult to manipulate because there's, I think, a lot more variables involved. Especially, I've looked at a lot of studies that
Hmm.
take a look at the difference in diet relative to disease accumulation, and
Hmm.
they're kind of vague to me because it doesn't really take into account a lot of information about the quality of the diet. Like
Yeah.
two different groups may have, different fat intakes, but it's also like what kind of fat, where's
Yeah.
that fat coming from? Is it, from plants, from meat? Like, because that
Hmm.
really will make a really big difference. And I
And
can
the
imagine
better
this a lot.
studies can tease that out. They have a large enough group that they can query them in detail about what they've been eating, and you can say, okay, we got these ones who are heavy duty um, red meat consumers, and these
Yeah.
folks who are vegetarians but eat a lot of fat.
I
Yeah,
want to a study
we
like that
can do
would be
that.
kind of cool. Yeah, if like a pure carnivore diet versus like a pure vegan diet, I would that would be interesting. But I can imagine rats, I mean, sorry, mice are a lot easier to like draw conclusions from than
Yeah, well, I mean, we
humans.
control their lives, their
Yeah,
little simple lives in their
yeah,
cages. Um, but there are things that you have to keep an eye out for. I'll tell you. Um, I don't want to go off on too many tangents, but
you're
I have
fine.
time to talk.
Yeah,
We had a set of experiments going. The current one, actually, that was published in Oncotarget, and we were surprised that our mice were starting to gain a little bit of weight, and um, it wasn't like they were um, extremely obese in a time frame that made a difference. Um, we found though that some of the animals were becoming rather obese in old age, so it made us wonder how reproducible our experiments are going to be. And what happened was they improved the living conditions in our animal rooms so that the temperature was better modulated and airflow is better modulated. Well, the mice were happier, they were less stressed, and when you gave them a high fat diet, they tended to gain a little extra weight. But then we discovered that most of them didn't really get fat until the very end um, of the dietary study when um, beyond the time when precancerous lesions would have formed, um, and beyond the time when they're really headed towards cancer. But to play it safe, what we did was is we examined the properties of the higher weight mice versus the lower weight mice in terms of whether BP3, for instance, on the low to high diet, whether BP3 increased incidence to the same extent in both skinny mice and moderately fat mice. And it turned out there was no difference at all in the experiment. So they were putting on a little bit more weight and, you know, didn't make any difference. We didn't think it would because all the weight gain was at the end, but we wanted to make sure that there wasn't a change in outcomes because of that.
right.
So yeah, you got to be nimble too in these in doing these kinds of work. You see something didn't turn out the same way, you got to go, oh, let's
Yeah.
design an experiment to see what might be going on here. We were able to do that.
And so the results that you kind of translated onto humans, as in like the conclusion that BB3 will kind of exacerbate tumor development in mice, it doesn't necessarily translate to humans will end up with a tumor once BP3 is in their bloodstream. It's more like it adds to the overall risk of
Yeah,
cancer.
yeah, that's what that would be our caution. That um, that it's just another insult in the environment, and um, if someone uses sunscreen containing BP3 oxybenzone or doesn't, is it going to make a difference in their risk? Probably infinitesimally small by itself, right? But when you consider um, the range of factors that we have in the environment that we're all exposed to, of which many we actually have choices we can make. Um, we can choose what sunscreen we do use. Um, we can choose what we eat. So clearly, eating a high-fat diet gives a different outcome than a low-fat diet. We can choose um, whether we use products that might have um, polyfluorinated compounds in them, um, which have other adverse effects. Um, so, you know, we I guess the hope is we identify all kinds of factors that are out there that incrementally increase our risk, and we first of all, um, through communication to the public, say, well, look, all these things show some risk in animal studies. You have to personally choose among the many things that are out there that have some level of risk, which things to avoid. We recommend avoiding as many as possible. Um, but we know that in this world, it's hard to avoid every possible
Yeah.
risk that we have in the environment. So um, I guess the idea is to inform people and educate them so they have more control in themselves about choosing what kinds of chemicals they subject themselves to. We don't know enough, I'd say, to say, oh my gosh, we should outlaw BP3 just on the basis of the study that I've published, although actually some places have, because they found, for instance, in Hawaii, they've outlawed sunscreen with BP3 because they found it actually can damage coral.
Oh, that's interesting.
And um, the coral can incorporate this chemical, and because it absorbs ultraviolet radiation, part of the chemistry behind that is um, it can generate toxic um, oxidative species in the metabolism of the coral that end up damaging the tissue of the coral, interfere with them being able to lay down the um, calciferous skeleton that they live in.
Right.
Um, so, yeah, it's pretty complex, the effects on all these things. And it's great. You know, some people who are experts on coral actually did some experiments in, there was one lab, they did some experiments in aquaria where they culture um, coral, and they looked at the effects of oxybenzone, and they found that coral development was hindered. And they've since found that the levels of in certain beach areas that are really popular in Hawaii, among other places, that the amount of BP3 in the water um, is really pretty high. You think, oh, it's an ocean, but in fact, because it's in shallows and there's not a very strong current in the shallows, and you got lots of people splashing around, slathering themselves with sunscreen. Um, um, you know, at peak beach time, there's a lot of this stuff, and some of the coral reefs are in protected areas. People are swimming there and exuding their BP3 into the water, and it's believed that it actually has some impact on the coral
There
in
was
the
a
natural world. Yeah.
there was a previous study that I saw on Oncotarget that saw that like some 98% of the population has BP3 in their system. Is
That's
that
right.
is that directly from sunscreen or is there another source?
Mostly sunscreen, um, but it's also in a number of personal care products besides sunscreens. Uh, it's been incorporated into lip balm, makeup, essentially anything that you want to protect from interacting with ultraviolet light. I even found it in my aftershave um, cream that I was using, and I stopped using it because I said, and that's part of it is, even if there's this level of uncertainty, if you um, see unexpected biological activity in something, why not avoid it if you can, if you have an alternative? That, and that's something a lot of us subscribe to, is this idea of a precautionary principle that even if you don't know with certainty what the exact risk is going to be, even though we do know that there are scenarios we can create in the laboratory where there is a real risk, we can't easily translate that level to the risk to humans. But what we can do is we can say, well, this is not inert. This is biologically active. If there's an alternative to use something that isn't biologically active, why not use it?
Yeah. During the we had a meeting previously on HPCAC, and you talked about how you used like I think was it zinc sunscreen that you
Yeah, personally, I
yeah
use either zinc or titanium oxides,
titanium
or
okay
many of those ointments come with a combo, and those are inert. They don't do anything.
yeah
They're just they tend not to be as cosmetically
please yeah
desirable because you have white streaks on your face.
yeah that's probably the main
But
reason why they
yeah,
use BP3
yeah.
yeah
But I'm at the beach. Do I care if I have white streaks on my face?
yeah. Have you looked at the effects of like phytoestrogens like
I haven't
the
done any experiments
no
on them. Um, the most the one that there's the most study and discussion of is genistein, because it's in soy, and
yeah
um, there's actually, it's interesting because there's literature that comes down on both sides of the story, um, that it might even be protective, although I guess the most recent conclusion is that it might have a mildly protective effect if you eat soy products your whole life, um, suggesting that there might be some um, effects of genistein in um, during puberty or pre-puberty that are beneficial. But I don't think people have a keen mechanistic sense. Against that, there have been studies that show that um, a lot of cancer cell lines, if you add this to their culture media, they grow faster. Um, so human epidemiology hasn't really come up with a definitive answer one way or the other. I think there's a mild protective effect that they've associated with ingestion of soy in um, Far East populations where eating a lot of soy is just a common component of the diet. And then people maybe take the logical leap that, for instance, um, immigrants um, from Japan who come to the U.S. and assume a completely U.S. diet show higher rates of breast cancer than those who haven't immersed themselves in U.S. culture. And yeah, that those data are real, but there may be other issues at play other than the amount of soy in the diet. It's very hard to be certain that, and the effect isn't huge either. I
so
mean,
in
it's another incremental
yeah
risk among the
yeah
many risks that we expose ourselves to.
I- i-
Um, yeah.
in what way would it be protective because is aren't like most hormone imbalances probably not a good sign
Yeah. Okay. So how could it be protective? It could be that Okay, this gets complicated. So we do know, and this is speculative, based on some things that we do know. Okay, but I don't know what the outcome would be in the real world. We know that various chemicals that can bind the estrogen receptor bind it a little differently than others. They have different levels of not only their avidity for the estrogen receptor, but the manner in which they activate the receptor is a little different. And the way you can imagine this is, okay, an estrogen receptor, it's a protein, right? And typically something that activates one of these receptors, it's a chemical and it binds in a pocket on the protein. It changes the shape of that protein that either allows it itself to bind to DNA and turn a gene on or off, which would be the case with estrogen, although it does that as a dimer of two proteins coupled together. Um, but another thing that happens when it binds and changes the shape, um, the regulation of genes requires not just estrogen receptor or any single transcription factor, but it generally requires several factors that bind in concert and end up talking to each other effectively. They touch each other and change their collective properties on the DNA and the outcome on the DNA. So, for instance, there are these factors that bind the DNA, and then they recruit what we call co-activators or co-repressor proteins that um, have activities associated with them, like um, acetylases, which is something that would add or take an acetyl group off of the histones that are binding to the chromosome and make those histones bind more strongly or less strongly and govern a little bit how exposed the DNA is and how open it is, which changes how strongly it gets expressed. And one chemical like genistein versus another chemical like estradiol can bind, and the shape of the protein will be a little different. How it contacts these accessory proteins that are also involved in transcription. We know that it could change the way they interact. And that's one way that one um, that a hormone, a natural hormone versus a somewhat chemically different um, surrogate for that hormone might give a different outcome. It's not just simply plus or minus, is something occupying the binding site or not. Depending on what binds in the binding site, the shape of the protein changes, what other proteins it can interact with to affect an effective um, transcriptional complex. One thing I'll say though is when that we do know is that if someone is suffering from breast cancer and let's say it's a receptor positive and they're going to take tamoxifen as a treatment. Tamoxifen is an analog of estrogen that can bind to the receptor and prevent its activity.
Oh, so it's just taking
It,
space.
yeah, it takes up space essentially.
Yeah.
And what they found is that genistein inhibits the activity of tamoxifen to inhibit cancer. So presumably what's happening is it's competing for the tamoxifen and it doesn't bind to the estrogen receptors effectively because they're all trying to bind to the same place and they all have slightly different properties. And there are studies that show that this tamoxifen treatment for ER positive breast cancer is less effective if somebody's on a high soy diet. So most physicians would probably advise somebody who's on tamoxifen therapy not to eat a lot of soy products.
That is that is
So,
complicated.
so
Yeah,
it's interesting too, right? It's not just a clear black and white.
yeah,
It's context dependent, right? If you're already sick with cancer, there's some cautions, extra cautions you're going to want to take that you're not going to really be worrying about if you're healthy.
yeah. So then, how much of a role does estrogen exposure play into cancer risk then?
Um, I don't know if we can parse out for sure. Um, we do know that excessive growth stimulation during puberty can have a bad outcome. We know that, um, for instance, that early puberty, having the hormones, the sex hormones, estrogen and progesterone kick in earlier, um, has a clear risk associated with it. So that's a little bit of a surrogate for abnormally high or lengthy exposure to estrogen and or progesterone. Um, so, okay, so there the logic is, just to draw it out a little more clearly, is um, a woman who has very early puberty has greater risk. And one of the things that we know occurs in early puberty, if it's precociously early, those are women who are producing estrogen and perhaps progesterone at an earlier time than the general population. They tend to have greater risk.
So because estrogen does play a role in fusing the growth plates with that, is
Oh,
that what you're
no,
talking about?
no. I
No?
mean, there are issues in other organs, but just in the
Oh,
mammary
okay.
gland, it's driving proliferation in the mammary
Okay.
gland.
Okay.
We know that, but another um, hormone. This is a protein hormone that's made within the body that can drive growth of um, mammary epithelial cells, which are the normal cells from which tumors arise, is a factor called IGF-1,
Yeah.
interferon-like growth factor 1. And high levels of that are associated with greater risk. And that's another factor that can simply drive proliferation.
Yeah. So I actually wanted to touch
We
on this.
worry about things that
The
just drive proliferation. Yeah.
Yeah, yeah. So like in red meat specifically, there are specific amino acids that could increase IGF-1 are and like mTOR and like growth hormones like that. Is that one of the reasons why like red meat might be associated with more cancer proliferation, just because
Hmm.
downstream it could increase IGF-1?
Um, well, I'm not really familiar with anything that has. Um, I haven't read everything that's out there.
Yeah,
I'm not familiar with a study that found that certain amino acids that are enriched in red meat are going to lead to more synthesis of IGF-1 or any other hormone. Maybe I just, I don't read everything. I don't have enough time
yeah,
to look at it. That would be. I'm a little dubious if it was as simple as that. I mean, one of the reasons that we like to that we started studying fat from red meat is that it tends to be enriched for saturated
yeah,
fatty acids and mono unsaturated fatty acids. And those are two fatty acids that we have our suspicions about. So in some of the earlier studies that we've published on diet, we used diets that were high in saturated fat and and mono unsaturated fat. And we did that by adding lard to the diet.
Yeah, that's what you
And,
fed the mice for this
and
study, right, Lard?
yeah, and you do that kind of a diet, and irrespective of uh, whether they get the lard in puberty or they get it in adulthood or they get it lifelong, they're going to have um, higher incidence of breast cancer. What's interesting in those studies is that when we fed the mice um, lard only in puberty, which for a mouse is from age 3 weeks to 10 weeks, um, we saw higher rates of breast cancer throughout their lifetime. So they're going back on a normal diet at 10 weeks of age, just as they enter adulthood, and we've put them on this healthy low fat diet. They're still going to have a higher rate of cancer than the mice that have been on a low fat diet their entire life. And so the mouse might live for 1.5 years, or we follow them for 1.5 years in these experiments. So spending 7 weeks on a high fat diet's enough to um, give them the kiss of death, so to speak, um, in terms of tumor occurrence.
Do you know like the direct mechanism that might cause like a higher fat diet to increase tumor rates?
Um, there are theories. I don't think we've gotten to the point where we can point to a precise pathway, but things we do know about fat, it increases. Well, it does a lot of things. Okay. Um, high fat diet changes the microbiota in the gut. Different bacteria are going to live in the gut depending on whether you have a high fat or low fat diet. And and the bacteria that grow on a high fat diet um, release more pro-inflammatory products. And even though fat may not directly initiate a tumor, if it creates an inflammatory environment in the area that a tumor is growing, that can be bad. Another thing that high fat does increases the permeability of the gut, um, so that various products that can induce inflammation can get into the bloodstream um, more easily on a high fat diet. Um, there's also there's a receptor that's on the surface of many immune cells that actually can bind um, a saturated fat, palmitic acid. And when it binds that, it initiates um, inflammation. It sends out signals that turn on genes that participate in inflammation. So probably I'm thinking mostly through inflammatory processes is how it's going to affect things. I mean, the successful research that's been published suggests that, yeah, it's likely through inflammatory processes is how it's increasing cancer risk. But there may be other mechanisms involved.
What is what do you mean specifically by inflammatory responses?
Um, so inflammation, whether um, it varies a little bit, inflammation depending upon various mediators, but um, typically what is going to happen, let's say an immune cell that's involved in mediating inflammation will make certain um, cytokines, which are smallish protein hormones that they secrete. A gene, like a common one that's been associated with breast cancer a little bit is interleukin 6. and interleukin-6 can be synthesized by macrophages, and they'll do it in response to a chemical that bacteria shed called lipopolysaccharide, um, which is it's a saccharide modification that you see on proteins in bacteria. Um, and what inflammation is, it's a set of signals which usually are saying, oh, there's been a wound at this part of the body, or there's an infection in this part of the body. Let's mobilize immune cells to go there and defeat any infection that might occur. Well, unfortunately, those kinds of responses have side effects. So inflammation is a good thing when it's dealing with an infection. It brings a bunch of immune cells into a localized area. It swells up because you have more uh, blood and serum entering a particular area, carrying hopefully lymphocytes that are making antibodies and T cells that are helping direct the lymphocytes to attack things with their antibodies, the B cells attacking the pathogens. And you have all this kind of stuff going on. But when an inflammatory response doesn't resolve, if it doesn't eliminate the infection, if it doesn't help heal a wound, and just sort of continues at a low grade over time, um, having these cytokines just floating in the microenvironment of um, cells that might be on their way to becoming a cancer cell, um, it can help them do things to grow as a cancer cell, um,
Oh,
rather than just be killed by some other arm of the immune system. So some of these inflammatory cytokines, they can promote vascularization. Um, they can inappropriately cause proliferation of some normal tissue. It thinks it's growing to heal a wound, but in fact, um, it, in a proper situation, that circuitry is going to help them do that. But if it's a cell that's already on the way to becoming a cancer cell, it may give them a mechanism to escape growth regulation. And as it does that, maybe it collects more mutations and then becomes totally uncontrollable in a tumor. And there you are with your life at risk. Um, does that help a little?
no, and that makes sense because I feel like I've heard inflammation a lot, and it's kind of become a little bit of a buzzword, like in the
Hmm.
specifically in the nutrition space. Like I hear a
Hmm.
lot of foods that are very like inflammatory, and it's just kind of used
Hmm.
like open-endedly, and I don't really understand what they mean by that. But
Hmm.
that makes a lot of sense. So then, were you saying something?
No, no, yeah,
Yeah.
that's fine.
So so is there like a specific type of fat that you would recommend after all this? Because you know that saturated fat is what's primarily in lard. Is that like would that be something to avoid, or would you go more for polyunsaturated? And have you looked into like omegas, like omega threes, sixes, and
Yeah,
nines?
there's some literature out there on omega-3s being beneficial and actually subduing inflammation.
Yeah, that's what I've heard.
So, you know, um, fish is good, for instance. But I don't know how solid the studies are where, whether anybody's taken, a population of 20,000 people who exclusively eat fish versus 20,000 people who exclusively eat beef and looked at the cancer rates. I mean, that may be out there, but again, it's impossible for me to read everything that's out there. And,
Yeah.
so there may be something like that out there, some kind of study.
There's there's some theory that like the Japanese live very long and their diet is very like rich in fish, and I think Okinawa as well. Okinawa is getting a lot of press because
Yeah,
they
there are a lot of very ancient people there.
yeah yeah. There's it's like
Like
it's not
centenarians.
uncommon to see people
Yeah,
overnight there.
yeah.
Yeah, and they eat a lot of fish, so
Hmm.
that might be something that's interesting.
Hmm.
Yeah. So then, are you working on anything right now?
What I'm doing right now, I'm heading towards retirement, so things are winding down a little bit. But um, what we're mainly focusing on right now is this study that I talked about, but we had a little bit of a glitch w with showing slides in my Zoom
Yeah,
meeting with you guys.
I remember that.
But so maybe the message didn't get across as well as it might have. But what we're looking at is rather than inducing cancer um, and seeing how um, BP3 influences outcomes of cancer, um, what we're doing is we're just taking normal mice that don't have any enhanced susceptibility to cancer. We haven't put a mutation in their mammary glands that gives them cancer, and just see how, because now we suspect that BP3 is an endocrine disruptor.
Yeah,
So we said, well, what will it do to normal development? And one of my students, she just graduated with her master's, she did this experiment where we saw, and this was a little bit of a surprise, but we did an experiment where we gave these normal mice BP3 and then we took it away from them. And my initial idea for the experiment was, let's see if BP3 has any permanent effects that they're going to eat BP3 and something's going to change. Some gene will be elevated. And if we take the BP3 away, maybe um, the level of expression of that gene will go down. And we say, oh, that's a target of BP3 regulation. Maybe it's a growth hormone that's a target. Maybe it's IGF-1, something like that. Well, what we found was that the interesting effects occurred not when they were ingesting BP3 in their food, but when we took the BP3 away. And we're trying to get a handle on what's going on there. And what it looks like is when we give the animal BP3 and then we take it away, when we take it away, the level of IGF-1 goes up. So they're getting a growth stimulus. When IGF-1 goes up, another protein cytokine hormone goes down, and the one that goes down is called um, rank ligand, RANKL. L stands for ligand. And um, that's a cytokine or hormone that regulates the differentiation of mammary epithelial cells. It tells them when a duct is growing to branch. In pregnancy, it tells them to form milk-producing alveoli. It, it, its most important role is in preparation for pregnancy, but it also, in adults, it has something to do with um, the branching of the mammary gland ducts. Well, that when we take away BP3 and IGF-1 goes up, rank ligand goes all the way down. And morphologically, what we see is the branching of the gland goes away. It suddenly looks like an immature,
yeah,
not very well differentiated gland. Well, that's you know, that's clearly, warning. This is really doing something to normal development. So you got to think about what's going on, um, let's say in an early teenage girl who's like slathering sunscreen on herself. Is it interfering with her mammary gland development in some inappropriate way? Um, so the other thing we saw in in these mice was that, let me give you a little background. So it turns out that for mammary gland development to occur, actually the immune system is involved in that. You need to recruit um, macrophages and eosinophils. I don't know how much immunology you've had in class, but various cells of what's called the innate immune system. They have to be recruited for the mammary gland to develop. They're involved in tissue remodeling. Essentially, they help the duct to grow through the adipose tissue matrix and form a gland, which it's a complicated engineering process that requires a lot of cells to help that happen. Um, if um, if those cells aren't available, the gland doesn't grow very well. It looks kind of simple. And that made us think, so what's going on here? We've looked at a number of hormones, protein hormones, that are indicative of the activity of the immune system. And what we found is when we take away the BP3, not only does IGF go up, pushing probably proliferation, and rank ligand goes down, which decreases differentiation of the gland. But we found that a lot of the typical cytokines that are indicative of immune cell activity of T lymphocytes, they go away. They just, their expression is way down. So that suggests to us that the immune system is being inhibited, and that's interfering with mammary gland development. Well, interestingly, another piece of work that I was a collaborator on from another lab that was part of the Breast Cancer and the Environment Research Program Consortium. They did a study, the professor for that, her name is Sally Snyder. They did a study where they looked at what BP3 would do to metastases, whether, and it seemed to promote metastasis of nasty tumors in their model. One of the things it did is it reduced the activity of immune cells at metastatic sites. So I think it might be immunomodulatory too. And one thing to think about there is, and we haven't, um, done a critical experiment. I don't think I'll get to it because I'd need to not retire and I need to stay at this till I'm 105 years old. But most of the cells within the immune system are responsive to estrogen. So anything that can mimic estrogen or inhibit estrogen is going to affect immune function.
Yeah.
So that's another thing to think about too, is that estrogen is kind of an immunosuppressant. So that means it's not only women's health, it's human health. Everybody's got an immune system, whether you're a man or a woman. Estrogen has an ability to modulate immune function. So it just highlights another worry. We're not going to have time to dig deep on this new area of what's going on in normal animals, but we'll get out a paper about the initial findings, and hopefully some people can run with it, showing that it's interfering with mammary gland development when we do the withdrawal of BP3 at that point, and that it's at the same time, it's showing signs that immune function has been altered. And of course, there's the question, how does taking something away cause an effect rather than just having it return to normal? So that leads to another question. Sometimes when you treat cells with some compound that alters the regulation of genes, we get what's called an epigenetic effect. And that's, it can, through the activation of these proteins that cooperate with primary transcription factors, that they change the chemical modifications of histones and other proteins that just form part of the chromatin structure. And they can create regions of the chromosome that are closed and won't express things anymore. And those changes in the DNA can carry over through multiple um, divisions of the cell without actually mutating the DNA. It's just that some region of the chromosome has changed. It can be shut down or it can be opened up, and it can enforce a change in expression of certain genes that will last over time. And something like that could be going on. So somebody should, could say, well, what's a typical thing we do when we're looking for epigenetic effects? We say, well, what's the methylation pattern of the DNA? Are there sites that have methyl groups on them and some sites that don't? And does it change when we do a treatment? And does that last over time? There are ways of studying the chromosome after these kinds of treatments and asking questions, chemical modifications of the DNA, chemical modifications of chromatin proteins. Is something like that happening in response to treatment? That would be another avenue to follow up with this to get at a mechanism. But anyhow, it looks like there are effects that occur in normal animals that disrupt development. And just like with the tumors, um, we saw different effects on different diets. And actually, for this disruption of mammary gland development, it was stronger in the animals on the low-fat diet than it was in the animals on a high-fat diet. So, it's hard to study what chemicals do in the absence of thinking about what they're eating at the same time as being exposed. And then it also um, shows that, yeah, you get more cancer on a high-fat diet than on a low-fat diet, but on a low-fat diet, there's some kind of disruption in a normal animal that maybe is irrelevant to cancer, but the mammary gland isn't maturing exactly the same way. So
So
there's a
there's a lot of
worry there too. You
nuance.
can't win.
Yeah, there's a lot of
Really,
yeah.
but
You can't
but
really
you
trade
get
off those
different
things too, right?
sets of effects, and I think there's a generally, when they're doing toxicological studies, they don't do them in the context of a range of different diets. They just go, well, okay, and I understand that. Experiments get crazy. You have so many different variables, and then that means more animals. It means a more expensive experiment. It means a harder to analyze experiment when you have multiple variables. So to get a simple answer, we like simplifying, but on the other hand, some of the things we've done have shown that you get very different effects just changing diet, and something to be aware of.
Yeah.
If it's safe on one diet, it may not be safe on all diets.
There's still a lot. Yeah.
Yeah.
You've got a lot of stuff going on then, because this is a lot of
Yeah.
this is very complicated, and it's hard to draw direct conclusions with this because there's so much nuance in some of
Yeah.
these things. Yeah.
So that's why I say that the biggest conclusion that I would take away from from these studies is that BP3 is not inert. It's not
Yeah.
simply something that absorbs UV. It has a lot of biological activity, and we should be cautious about something that we tell everybody to use,
Yeah.
particularly in the summer when there's a lot of sun exposure. We tell everybody to use this. Um, and when there are alternatives that are not biologically active, why take this path? Why not use the safe ones, even if you have white streaks on your face?
Yeah. that study really like it brought like measurable results that yes, BP3 is having biologically active effects and we
Mhm.
should be wary of them. And I think that's probably the
Yeah,
most important
yeah,
conclusion out of all of them, right?
yeah, yeah. That's and the drug companies would say, oh well, it doesn't do anything. Well, you know.
Yeah. Have any of you guys tried to kind of alert like the source, like specifically drug companies, and
Um,
get them to
we
hopefully.
just publish and we let the world respond. You know, so when we published the work, we did a press release um, that said that it it was a compound that, at least in mice, it increased cancer risk, and we got some public response. I mean, we actually got some guy who was I think he was somewhere higher up in some dermatological society, contacted us and said that we were perhaps doing a disservice because people would stop using sunscreen because they'll be scared of it and then they'll get skin cancer. And all I could do is email and say, well, they could use other sunscreens. They're out there. It's not like you can only use this. So you get different responses from people on these kinds of
Yeah,
things. At one time, I did a presentation on the effects of high fat diet on breast cancer incidence. And another scientist at the conference um, commented at the end of our talk. She says, well, I think you might be doing a disservice by making people feel if they eat too much fat, they'll get cancer. Um, and it will promote young women um, towards bulimia, you know, and I didn't know how to answer that. I mean, to me, that was like crazy. Um,
that's a pretty far-reaching conclusion. Like you're just
yeah,
you're just you're just trying to find results. Like that's not necessarily what you find is a little bit closer to the truth. And if the truth, I don't think the truth's going to lead to that, right? Like
yeah, and I try to be nuanced. Like when we made a release, we said we're not saying this is essentially said in our press release that this says it's biologically active, and we need to be cautious about things that are biologically active that we're not aware of their biological activity.
yeah,
Yeah. And I think most of us who do bench science, we try to be really cautious because our own standards of proof are actually pretty high.
yeah,
A- and actually when I had to write the press release, I write something and then you give it to the publicity person who puts out the university press releases, and they take it and they made the conclusions far stronger than I would have. They said, well, people shouldn't use sunscreen because they will get breast cancer. I said, no.
yeah,
It's an incremental risk, and what
yeah.
we're saying is it's biologically active. So if you have a lot of choices to make, don't use this sunscreen, use another sunscreen. You it's just because they wanted it to sound more consequential and to be a sexier kind of
Yeah.
message than it really was. They, you know, because their job is just, hey, let's put ourselves on the map. Let's tell them that Rich Schwartz discovered something that's going to change the whole world of breast cancer. They, and I'm going like, no, this is important, but it's not quite important in the way you want to publicize it. So we always have that debate in messaging. It's something that we deal with.
That's something that happens a lot with studies that get, even if it's a little bit closer to this very pervasive idea, they will kind of market it that way. You see
Yeah,
that in a lot of studies where they'll draw direct conclusions from things that are a little bit more ambiguous. Right?
yeah.
So,
And I think, I where they are, the publicity folks if you work for the university, your idea is to publicize the research your university is doing to
Yeah,
put your place on the map.
yeah.
And it's easier to do that when it's a simple story where people can draw fairly simple conclusions. They're not into nuanced messages. And what we generally do as scientists, we try to keep the nuance into this. Because I don't know, truth is important. I mean, I think to all of us who do science professionally, and um, we're into accuracy of messaging, we're into um, truth is a big deal for us, I guess.
Yeah.
That's we enter this field because we for whatever crazy reason in our psychological makeup we have discovering new knowledge is important. Truth is important to us in whatever field we've chosen. We want it. We want our work communicated really accurately.
So how did you find like this field specifically, oncology and cancer research?
For me, um, it's kind of a long and winding road a bit. So I found that I was really excited by the biological sciences when I was a young guy. And the focus of it shifted. I had decided I was going to become a professor in the life sciences when I was like in junior high school. If someone had asked me, "What do you want to be when you grow up?" I would have said, "I'm going to become a professor." And then my interests, it cancer was interesting. Um, Neurobiology was really interesting for me. Um, Differentiation, how a stem cell becomes a specific kind of tissue was very interesting for me, those kinds of issues. Um, then when I went to graduate school, um, because because it was at that point, I guess I was really interested in tissue differentiation a lot and how the immune system develops from a stem cell into an active cell that can perform a health function. Fascinated me. And I worked in an immunology lab. And at the time, one of the things that we didn't have a clear picture of was how antibodies are expressed by B cells. But what does the gene for an antibody look like? I mean, at that point, we didn't even know what it was. So it's involved in those kinds of studies about 40 years ago. And then after I got interested in something that wasn't very health related. I found some peculiarity in the way immunoglobulin antibody genes are expressed that suggested something about how messenger RNA is spliced. I don't know if your biology, if they talked about splicing, but
A
it's okay. I
little
mean,
bit, yeah.
I don't know at what point these days they introduce certain concepts, you know. But essentially, what happens is when you make a gene product, you get a long strand of RNA that gets translated into protein. Well, it turns out that a lot of genes, or most genes, in fact, the coding information for the protein is not continuous, and pieces of DNA have to be cut out and the protein coding material spliced together to get something, a piece of RNA that can be read into a protein. And I found that there was sometimes that went awry, and you got an antibody RNA that didn't read through properly, and you got a defective antibody. So I got really interested in how splicing, this putting together of coding regions in the RNA, came together. So after I got out of grad school, I did a postdoc looking at splicing mechanisms. And certain experiments didn't work, and my advisor said, well, why don't you try this? This is a good model system. It's not what you're excited about, but um, so it was, I worked on how transfer RNAs can be spliced because there were good reasons to do it. But it was just pure biochemistry. And I did some pretty successful work in that area, but then it didn't float my boat. I really, in the end, wanted to do things that had obvious health impact. So I quit that area after 3 years and went to yet another lab for another postdoc where I worked on lymphoma and leukemia and looking at how the activation of oncogenes affects that. So I did postdoctoral work in there and published some nice papers, and then I became a professor working in that area, and I pursued that for a few years, and then um, I hit a roadblock in funding, even though we had some nice results, but um, things sometimes I wasn't going in the right direction. Because you've got to have work that excites people so they can give you a lot of money so you can do
Yeah.
these experiments. So then I said, oh, well, here's an interesting thing. I found that in some of the leukemic cells that I was studying, something that started out as a tumor of B cells, an antibody producing cell, suddenly took on the properties of a macrophage. It had switched its differentiated phenotype in the course of its tumor progression. And I said, oh, that's really fascinating. I'll study that. So I got money to study that. And I did that for a while and got a whole string of papers. And it led to understanding how certain transcription factors regulate the inflammatory products that are produced by macrophages. And then a colleague of mine got interested in some of those transcription factors that were regulating inflammation because they also regulated mammary gland development, which isn't surprising in a way because, as I told you, a lot of the immune cells are important
Yeah.
for allowing mammary gland development to occur, a lot of the same factors. So we were collaborating on some stuff that was related to mammary gland development. And then as those experiments went well, I got more and more interested in that and decided, I'm going to go in this field because it's expanding and there's good funding to do good experiments and kind of switched gears. So then I worked on that, and that project ended up getting funded by the Breast Cancer and the Environment Research Center. And we were looking at the effects of diet on cancer. And then when that grant ended, the people who ran that program, they said, we love your work, but we're really focusing more on chemicals now. We're not as interested as an institute in dietary issues. We'd like you to. So we said, well, what if we looked at the interaction of chemicals with diet? And they said, yeah, that works. So then the next grant was BP3 and high fat diet. And so I just briefly told you about my about at this point about a 50 almost 50-year career switching from one area of research to another. And I think for a lot of us, we reinvent ourselves periodically in terms of the science that we want to focus on. Part of it is our changing interests. Part of it is to do good work, you need adequate funding. So you look to see what do people care about and what are you interested in, find the intersection of those areas and develop research projects. So that's how I got involved in benzophenone 3.
Okay.
The folks at the
So
National Institute of Environmental Science said, find a chemical. So we did.
okay, so I in the email I told you it would be half an hour, but we're almost at like an hour and
Yeah,
a
I
half now.
enjoy, we're over that, but
Yeah,
you can tell I enjoy talking to
yeah.
people who are interested in the work, and
This was really fun.
that's good.
I
I
definitely want to do
enjoyed it.
more. Yeah, I definitely want to do more research, specifically into the life sciences. I just think this is interesting. I just think the human body itself and like life is such an enigma. Like there's so many unanswered questions. It feels simple at a surface level, but when you look into it, it's pretty interesting.
I
So
think, for me, one of the transformative things in my career was when I was, as I was finishing up my sophomore year in college, I, I um, went around and found a laboratory that would take me and give me a chance to just do experiments at the bench and under some direction. And that sort of really set me on a career path, having that experience in a functioning laboratory. So, so that's how I kind of knew where I wanted to go, but having an experience in a lab when I was 19 years old, starting at that point, um, confirmed for me that I was on the right path. Because I thought about, oh, am I going to be a research professor? Do I want to become a physician? Do I want to, um, and then I had other interests. Did I want to get into, I thought about all kinds of stuff, creative writing, um, political science. I had a pretty wide, I mean, I even spent, while I was in grad school getting my PhD in biochemistry, I spent a year volunteering at the student radio station. I had a music program with a friend, and we DJ'd a show that was a mixture of jazz and classical music. So, but you know what? You got to pick something at some point
yeah,
and just roll with it. And and the fact is, you can stay interested in things other than your primary career, although it can be quite consuming. But you don't necessarily have to give up everything else.